Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive, where we unpack the science behind landmark research papers. Today we're tackling a real giant, Fire and Mello's 1998 Nature paper. And let's just jump right in with the absolute shocker, the thing that turned everything on its head. For years, people thought if you wanted to silence a gene with RNA, you'd use a single complementary strand antisense. Kind of like a mask. Simple idea.
Yeah, simple, but it barely ever worked reliably. It was messy.
Right, and then Fire and Mello come along and basically say, nope, you're looking at the wrong molecule entirely. They discovered that double-stranded RNA, dsRNA, wasn't just involved, it was the actual trigger. And not just a trigger, it was incredibly potent. Way, way stronger than single strands.
And the truly wild part, you needed almost none of it. We're talking maybe a handful of molecules per cell to completely shut down the gene.
Which just blew the whole masking idea out of the water. This wasn't just blocking. Something active, something catalytic had to be going on. So our mission today, unpack this paper, potent and specific genetic interference by double-stranded RNA in Caenorhabditis elegans. How did this simple experiment in a tiny worm kickstart the entire field of RNAi?
It really is hard to capture the frustration back in the mid-90s. The potential of using RNA was obvious, but practically speaking, it was, murky is a good word for it. People were really struggling. Everyone was focused on antisense. The logic seemed sound, Gene makes messenger RNA, you add the mirror image strand, they stick together, and the cell can't read the message anymore.
Like putting tape over the words.
Exactly, simple concept. But the reality was flaky at best. Sometimes a tiny effect, sometimes nothing.
That's it. Totally inconsistent. And then you had these weird results coming out of plant biology, this thing called co-suppression.
Right, where adding more of a gene could somehow switch off the gene you already had. Makes no sense intuitively. Exactly the opposite of what you predict. So hints were there. Hints that the cell had some kind of internal RNA-based defense or regulation system, but nobody knew the trigger. Was it the sense strand, the antisense, or maybe the two stuck together, the duplex? That was the big question. What's the actual active form?
That's the puzzle they set out to solve. And they chose C. elegans, the nematode worm. Why the worm? Why not something else?
Oh, the worm was perfect for this. It's transparent, so you can literally watch development unfold.
Okay, yeah.
The genetics are super well mapped. But the killer feature for this work, you inject an adult worm, and that injection material gets into both the body cells, the soma, and the germline cells, the ones that make eggs and sperm.
Oh, wow. So you can see effects in the parent and potentially its kids.
Precisely. That direct link from injecting a molecule to seeing a clear effect across the organism, even into the next generation, was just invaluable for clarity.
So that leads to their experimental setup, which was just elegant, super clean. They basically forced the issue. Sense, antisense, or both together. They picked genes with really obvious effects, right? Like UNC22.
Yeah, UNC22 is a classic. You silence that, the worm twitches uncontrollably. Can't miss it under the scope.
So group 1 gets pure sense RNA. Group 2 gets pure antisense RNA. Group 3 gets the two mixed together beforehand, the double-stranded stuff.
And the results, bam, crystal clear, just laid out right there in their initial findings. It basically showed years of effort had been slightly off track.
How different were the results?
Oh, night and day. The single strands, sense or antisense alone, almost nothing. Maybe a tiny, tiny hint of an effect, like one worm twitching slightly.
Okay.
But the dsRNA mixture, whoa. Massive effect, potent, specific silencing. You got worms twitching violently or even paralyzed, depending on the gene. It worked, and it worked incredibly well.
So just like that, they showed the trigger has to be double-stranded. That moved silencing from this weird, unpredictable art into something with actual rules.
They found the key, the structural requirement. But honestly, the next part was maybe even more stunning. It wasn't just that it worked, but how far it worked.
Right, because they injected it in one place, but the effect wasn't just local, was it?
Not at all. This is where the worm system really shines. They showed this silencing signal wasn't just staying put in the injected cells.
Okay, so looking at their analysis, they saw the effects in the worm they actually injected.
Yep, the adult animal showed the phenotype.
And in its offspring, the next generation.
That's the kicker. The effect was heritable,
Yeah.
which means the dsRNA signal, or something triggered by it, must have spread throughout the adult worm systemically and gotten into the germline to be passed down.
That's kind of mind-blowing for developmental biology. You're essentially passing on an acquired trait via an RNA molecule.
It really is profound, a molecular signal crossing tissue barriers, even generational barriers. For it to be that pervasive, the underlying mechanism had to be serially powerful.
Which brings us to the mechanism itself and the dose. How much dsRNA did it actually take?
And this is where it gets really interesting, moving from what works to how it works. They did careful titrations, measuring how much dsRNA they put in versus how much the target mRNA, like MEX3 mRNA, disappeared.
So the big reveal, how little do they need?
Almost unbelievably little. Their data showed that just a few molecules of dsRNA per cell were enough to get strong silencing.
Okay, let's pause on that. A few molecules, like single digits, 10, 20.
Yeah, in that ballpark. Maybe tens of molecules per cell.
But a cell has, what, thousands, maybe tens of thousands of copies of the target mRNA it's trying to silence?
Exactly. So if
Yeah.
you inject, say, 10 dsRNA molecules and they need to silence 10,000 mRNA molecules, well, those 10 aren't just sticking to 10 targets and calling it a day. That math doesn't work.
Right, that kills the simple blocking idea, the stoichiometric model. You'd need way, way more blockers than targets if that were the case.
Precisely. You'd need at least one blocker per target, probably more. Seeing potent silencing with just a tiny input fraction meant something else was happening. It strongly argued against simple one-to-one masking. It meant the dsRNA wasn't the blocker itself, but more like a key. A key that turned on some kind of cellular machine, a machine that then actively hunted down and destroyed all the matching mRNA targets.
So the dsRNA molecule is just the trigger, kicking off this massive amplification cascade inside the cell.
Yes, a catalytic or amplification component. The dsRNA acts like a guide, telling a powerful pre-existing cellular system what to destroy.
And the thinking now is that this system probably evolved to fight viruses, right? Because viruses often make dsRNA.
That's the leading theory, yeah. It's likely an ancient antiviral defense mechanism. Fire and Mello basically figured out how to hijack this cellular weapon system for targeted gene knockdown. It's brilliant.
So they didn't invent something new, they uncovered something fundamental that shifts it from just a neat lab trick to a basic biological process.
Absolutely. It was a paradigm shift. The paper fundamentally established that cells have this built-in enzyme-driven pathway triggered by double-stranded RNA. And that hint of amplification. It directly pointed towards the existence of the molecular machinery we know about now, enzymes like dicer, which chops up the dsRNA, and argonaut, the enzyme that actually slices the target mRNA.
And the practical impact for labs,
Yeah.
for functional genomics.
Immediate and transformative. Before this paper, knocking down a specific gene was laborious, often requiring complex genetic engineering that took years, and often it just didn't work well. After 1998, suddenly you had a relatively simple programmable recipe. Make the dsRNA for your gene of interest, inject it into a worm, and boom, you could observe the loss-of-function phenotype reliably.
That must have changed everything for C. elegans researchers almost overnight.
Totally. It enabled the first large-scale systematic screens where you could knock down basically every gene in the worm genome one by one and see what happens. That was unthinkable before. And it didn't stop there. This work is the direct ancestor of the dsRNA tools used in pretty much every molecular biology lab today across all kinds of organisms. And it paved the way for RNAi therapeutics being developed now. It made functional genomics much faster and more accessible.
So the huge takeaway. Fire and Mello took gene silencing from being this unpredictable, almost mystical thing and turned it into a defined, principled molecular process, all by identifying dsRNA as the key trigger. And you have to credit the environment too, right? The collaboration between Carnegie and UMass.
Definitely. That combination of really careful RNA biochemistry, making sure those single and double strands were pure, coupled with the power of worm genetics, was crucial. It gave them that undeniable clarity.
The results were just so clean, so reproducible, the field could just grab it and run with it immediately.
Exactly, which is why it spread like wildfire and, of course, earned the Nobel Prize. But okay, let's leave you with something to chew on. connecting this amazing discovery to challenges that came later. We've established this RNAi process is incredibly potent. Just a few dsRNA molecules trigger massive destruction because it's catalytic amplified. It's a powerful internal weapon.
Right.
So if the cell evolved this super powerful system, presumably for defense, what kind of control mechanisms do you think it also had to evolve to keep that power in check, to make sure it doesn't go haywire? Think about it, because when researchers tried using this in other systems, especially mammalian cells, they ran into problems like off-target effects, the machinery accidentally slicing the wrong mRNAs, and also triggering the innate immune system, which is specifically designed to freak out when it sees foreign dsRNA. How does the cell normally manage that fine line between potent defense and, well, self-destruction? Something to ponder.